HIV, also known as the human immunodeficiency virus, is a type of retrovirus that infects humans. Over time, it causes AIDS, acquired immunodeficiency syndrome, a condition where progressive failure of the immune system due to too many helper T cells being killed occurs. This can mean life-threatening, opportunistic infections and cancers can kill the host. So, what is a retrovirus? Well, first of all, we need to look at the structure of one. If we look at the structure of HIV, which is a retrovirus, we can see that the core it contains is genetic material, RNA, including some proteins, which also includes the enzyme reverse transcriptase, which is needed for virus replication. There's an outer coating of protein around these called a capsid. This is actually what's injected into the host cell. An extra outer layer called an envelope is also included. This is usually a membrane stolen from the cell membrane of a host cell. Then, sticking out from the envelope are copies of attachment proteins which help HIV attach to helper T cells receptors. You can see that HIV is a retrovirus because the genetic material it holds is two single chains of RNA, which is what makes it differ to become a retrovirus. In terms of replication, HIV's attachment protein binds to the CD4 receptor on the helper T cell. This CD4 receptor is characteristic of the helper T cell, which is why HIV can only infect helper T cells. The attaching of the attachment protein and spike protein brings the virus towards the cell and fuses its envelope with the membrane. This then leads to the virus releasing its capsid into the cell. The capsid is then digested by enzymes, leaving the RNA by itself in the host cell's cytoplasm. Once in the cytoplasm, the reverse transcriptase is then used to make two complementary strands of DNA from the two viral RNA templates. These can join together by hydrogen bonds by the base pairs to make a double-stranded DNA. Now, the double-stranded DNA is inserted into the human DNA in the nucleus. This may be done by the enzyme integrase. Then, host cell enzymes are then used to make viral proteins from the viral DNA found within the human DNA. This is replication of RNA for more HIV viruses, and host cell enzymes are also then used to make more reverse transcriptase enzymes and other proteins needed for the HIV virus. This part is part of a lytic cycle as more parts of the virus are replicated. Now that enough HIV components have been assembled, the new structures go back to the surface of the cell where the spike proteins have been. After many HIV viruses are replicated, they bud from the cell, take the host cell's membrane with them. This is another part of lytic replication. In comparison to lysogenic replication, the virus would remain in the cell. As you can see, the HIV virus takes the host cell's membrane as it buds from the cell. These newly replicated viruses can now infect other helper T cells, and the process repeats. This is how this can eventually progress onto AIDS, where enough helper T cells are killed. Unfortunately, there is no cure for HIV. The best thing we can do, however, is use HIV treatment drugs, which we can call antiretroviral drugs. Some of these drugs can be inhibitors to the enzymes. This can be in areas such as the initial attachment of the spike protein on HIV's envelope. As you can see, the attachment protein is inhibited, and therefore the virus cannot enter the host cell. There can be other areas such as stopping the replication of complementary DNA strands, or by inhibiting integrase, an enzyme which said earlier can carry the DNA to the nucleus to be put into the host cell DNA. However, with the inhibitor in place, it cannot join onto the DNA strand, therefore preventing the HIV genetic material to join onto our host cell DNA. Lastly, it is also known that antibiotics cannot treat viral infections. This is because antibiotics target foreign mechanisms and structures that aren't found in the body. Looking at the diagram, there are many structures in bacteria that is not found in the body. However, a virus has components that are not foreign to the body, therefore antibiotics cannot be made to attack it. It would not work to use drugs that target virus structures because, since they are found in our body, they would target our own cells and would kill them. Hello guys, thanks for watching. Just to mention importantly, a lot of the animation in this video is only fundamental information relative to the A-Level AQA specification. There are lots of other videos that are helpful to watch if you'd like to go into more detail, but anyhow, this was so much fun to make and it took me a few days to put together, so I really do hope you enjoyed it and found it useful and informative. Thank you.